Horizon Discovery is a genomic translational company, and we have expertise in genome editing, discovery services, and diagnostic reagents. And the diagnostics reagents is what I'm going to be focusing and talking a lot about today. So the question that we asked ourselves 2.5 years ago when the idea of the business unit was in its infancy was, how do you know that your molecular assay is working today? And the common response and the common answer is that the positive control that you're running gives you the expected result, and likewise the negative control that you're running gives you the negative result as well, or the blank result. And so what we then ask next is really what are the controls that you're using, where are they from, and what is their suitability and how they're validated? Because it's often the case that certainly the kit controls that are used have been manufactured specifically with that kit and gone through all of the developmental steps and stages, so that when you then get to using that kit, the true reason for that control is to show that the components of that kit work. You can't use the controls for the kit necessarily to actually test or validate that the samples you're running will be compatible with the kit that you're running. And that's where reference materials and reference standards really play an important role. So here's a simple schematic of where a tumor sample goes when it's either goes through histology for diagnosis and therapy, or some FFB sections will go to the molecular pathology or molecular diagnostic lab, where it will be the DNA will be extracted and then genotyped. Now, throughout this whole process, there's opportunity and for variability to actually creep in. So this one part will be the sample quality, the sample heterogeneity, DNA quality, platform variability, and mutational characteristics. And what I'm going to do is talk through each of these points on the next 5 or 6 slides. So, tumor sample heterogeneity. We all know that human tumors do display a startling amount of heterogeneity, and this can be either driven morphologically or by epigenetics, but a large proportion of this will be driven through the genetics and divergence at the genetic level. And here's a schematic where you have a wild type sample all the way through to a tumor sample which has got 100% tumor cells. And this is either at the phenotype or if you're looking at the phenotype level, you may have 100% phenotype, but then when you actually go down to the genotype, depending on whether the mutation that you have incorporated is either homozygous or heterozygous, will then govern whether or not the genotype that you have that you can detect is at 50% or 100%. And so if you're working at the level where you have a 10% mutation, you will require a limit of detection of at least 5% to be able to detect this if it's a heterozygous mutation. The next variability that needs to be considered is the sample quality, and this will be very much governed by the formalin fixation duration. So when you have when the biopsy or the resection surgery is taken, if it was taken, say, on a Friday afternoon, it could then be fixed for a full 24 hours, or it could even be over a weekend, a 72-hour fixation if it's not processed through the FFB straight away. And this would then lead to your sample being more difficult to extract the DNA or RNA if that was what you're looking for, and that would then really govern and also influence the quantity of DNA you can extract from your sample, the concentration and the purity. To also play into this, you have a number of different DNA extraction methods available, and these, you can get them from QIAGEN, from Promega. You can have homebrew methods, and all these different kits have slightly different chemistries, and that will then provide you with DNA with slightly different levels of quality in terms of the DNA quality. Now, one thing that we've seen, and it's actually often overlooked, but it's a very important step to consider in the process, and this is the DNA quantification. And that's because the most common method for measuring DNA content is nanodrop, and that is accurate when the concentration is 10 nanograms per microliter or above. However, when the concentration is below 10 nanograms per microliter, the best methods and the gold standard methods for testing the concentration are either picogreen or quantifloor. And I've just put together an example, and this is data from our lab, where we generated a concentration of 2.5 nanograms per microliter in a sample. And what we wanted to basically show was if you had a genotyping assay that required 20 nanograms to run, if you had a starting concentration of 2.5 nanograms, you'd have to add 8 microliters to get the required amount of DNA to put into your assay. However, and this is what we did, when we quantified using nanodrop, it's actually quantified at 6 nanograms per microliter. And then so taking this value, would only put 3.3 microliters in, and actually this would only give you a total of 8.3 nanograms for this particular assay. And so this would then lead to the assay being suboptimal. And if you were at your lower end of your limit detection, this would actually lead to you actually getting a false negative result. And so it's absolutely critical that the DNA quantification prior to loading your sample into your assay is accurate. Now, as I'm sure you're very well aware, there's multiple assays that you can use. There's obviously the homebrew methods that are often very sensitive. There's QIAGEN Therascreen. Entrogen have a number of KRAS and BRAF kits. The Vienna Lab strip assay. These, from data that we've seen from people we're working with, have a limit of detection in the range of 1. Secronome, the Roche Cobas system, pyrosequencing, that would have the range of about 5%, and then Sanger sequencing is typically accepted between 15% and 25% limit detection. Now on this slide, what was interesting is when we sent some of our standards out to one of our partner labs who were running Sanger sequencing, there's actually it's actually a very nice data set that highlights that different mutations and different primer sets will give you different limit of detections. And what we were able to highlight at this lab was that although for the KRAS G12A, the PI3 kinase H1047R, and the BRAF Fusion they had good sensitivities of 5% or 10%. For the PI3 kinase E545K, they had a limit detection 25%, which would mean that they would need at least a 50% tumor sample to be able to detect this mutation if it was heterozygous. And so in this case, what we were actually able to do was, once we'd highlighted this, they were able to redesign primers and get a much more sensitive assay set up. So just coming back to what I asked at the start of the questions I put at the start of the talk, with all of these variabilities that are coming in to the process, it's absolutely vital that the controls that you're using are very well validated and suitable. And we've spoken a lot to the proficiency schemes and the proficiency testing groups and have very good relationships with them. And they told us, and continue to tell us, that existing reference materials are very difficult to source. They're often genetically undefined and can be very variable. You've got your synthetic oligos, which are, will always be very easy to detect because they're so short, and for any of these assays, it's just a simple PCR to see them. The cell line mixtures have been very popular in the past, and the next couple of slides I'm going to sort of introduce why they need to be used with some caution. And then, obviously, I mean, primary tissue samples will at some point always need to be involved in a validation of a molecular assay. It's just the tissue with primary tissue samples, you don't want to be having to use them for the initial setup or development, especially if it's a rare mutation and the tissue samples are very difficult to come by. What you want to be able to do and achieve is get your assay up and running, understand the limit of detection, and then run tissue samples through the system to show that you can then detect real patient samples with your molecular assay. So here's some data from one of our partner labs, and this is where they came to us and told us that with they've been using an EGFR E746 A750 deletion cell line, and we're actually able to, using Sanger sequencing, they had a limit detection of 2%, which is was incredible, and much lower than you would expect. And so we asked them just to send the samples to us, and we would validate the samples they were using with our digital PCR platform. And at this point, you'll have to, there's more digital PCR data later in the talk, but you have to take my word for it that with our digital PCR platform, it is very sensitive and will give incredibly accurate results. And so what we did was we took their dilutions where they'd come out at 1%. They thought they'd made 1%, 2%, 5%, 7.5%, 10%, and 15%, and then the cell line undiluted. And we ran it through the system. And what we found was that what they claimed was a 2% dilution was in fact actually a 14% dilution. And this then explained exactly how they were able to detect what they thought was a 2% in using the Sanger sequencing method. When we then looked into this further and investigated the cell lines, we actually, we took their cell line, which is HCC827, and that's, we've called it source 1, and we also took the wild type that they were using, HeLa, and we did a copy number assay and looked to see how many EGFR copies there were. I'll start with the mutant. So with the mutant, there was 14 copies of the EGFR, which explains the much higher allelic frequency than expected. And what we then did as well was we brought in a cell line which was also was classified as HCC827 from a different source. And when we actually tested the copy number of that cell line, it came out at 38. So from this, not only can cell lines give you mutations with unknown copy numbers, depending on where the source of the cell line is, could actually give you introduce any variability. So although one cell line might be characterized by one lab, unless you get that actual cell line from them, if you get the same cell line from a different source, there may be variability being drawn into the assay. And what was also interesting was the HeLa that they gave us a copy number of 2.6, and then also when we got a second HeLa in from a different from the source 2, that was 2.7, and this is indicative of a heterogeneous population where you have a number of HeLa cells that have a copy number of 2 across for EGFR, and then a number of more cells that have a copy number of 3 across the EGFR locus. And so even if the copy number of the mutant was correct, actually mixing with a heterogeneous population would have thrown up errors in the limit errors in the dilution curve anyway. So there was a number of factors actually impacting on why this dilution curve was so messy, essentially. So what we set about, and this was in January in 2011, and what we continue to strive for, is we wanted to make reference standards that contained a defined copy number, a defined allele burden. They showed genome heterogeneity. They were either available as DNA or as FFPE, and they mimicked tumor genetics. And what I'm now going to talk through for the rest of the talk is how we have achieved this goal, and how we set about doing that. So it's probably a good time just to define what we feel a reference standard is. So it's an independent or externally validated reference material. It allows you to understand the sensitivity or the limit of detection of your assay, and it will facilitate you to monitor and maintain that the reproducibility of your assay is suitable for your application. And for kit developers, you can use it. You can use it for research and development. And this is where I can actually close the question that I put out at the start, in that a reference standard will tell you that your assay is working today, and that's the most important part of a reference standard. So, because at Horizon Discover we have the expertise in gene editing, and we used our gene editing platform Genesis to develop all of our reference standards. So what do we do along this process? This is a very simplified, very simplified schematic of what we do. So we take our what we would call a wild type cell line from like ATCC or other sources. We do a single cell dilution. This will then ensure that we have a clonal wild type cell line banked, and then we do extensive cell line validation on this wild type. Once this cell line validation is complete, we will then do the gene engineering and gene editing event, which will then ultimately create a clonal mutant cell line. And again, this will go through the same cell validation process. It's important to mention, so with our Genesis technology, we always introduce and create a heterozygous mutant cell line. And also, if you would like to have more information or are interested to know a bit more about the mechanics of the technology, this can be found on our website, and we have webinars where we have a full hour devoted to how the actual technology works, that will be available for you to log into. So cell validation. What we do is we confirm the identity of the parental cell line. This is through STR as well as running SNP6. We confirm that the mutation has been integrated at the correct locus using DNA-specific PCR and tang sequencing. This is comprised of somewhere between 8 and 12 different PCR reactions to ensure the integration. We then confirm the expression of the modified allele by running cDNA PCR and tang sequencing, and we confirm. And this is where we took a big leap forward last January, in January 2012, where we brought in our digital PCR platform and now using it routinely to confirm the clonality of our cell lines, as well as also confirming the gene copy. And the end point of the engineering is that we have we have the original clonal wild-type cell line, as well as our clonal heterozygous mutant cell line. And essentially, these two cell lines are identical, except for the one mutation that we've introduced at the endogenous site. This is a list of all of the mutations that we've currently engineered for our molecular reagents and quantitative molecular reference standards range. In total, Horizon Discovery has engineered in the region of 450 to 500 cell lines. And so we had the cell lines. We had the mutant and wild type cell lines that were isogenic, and we then went about generating two different types of standards. So one is our FFB Reference Standard, and the idea of that is to monitor and evaluate the DNA extraction process, as well as being able to genotype it through this workflow. And then also we have genomic reference standards, which can be used to understand the sensitivity and can be used for batch testing of the platform performance. This is just very simple, a simple schematic of how the different ways we actually make the standards. So the genomic DNA reference standards, we grow up cell lines, we extract the DNA, and we either provide the DNA as 100% mutant or 100% wild type, or alternatively we can create mixtures, and then you can have a mixture of the DNA that will then verify using digital PCR, and the mixtures are range from say 1% to 5%, 10%, depending on the need for the testing. On this side we have how we do the FFP. So we grow our cell lines up again. We then will pellet, formalin fix, and then go through our FFP processing step. If we want to generate a mixed FFP block, we'll mix the cells prior to formalin fixation, and then we end up with the mixed sections. So this slide is a very nice slide just to summarize what we've done with our genomic DNA reference standards. So this is using the digital PCR platform, the Bio-Rad QX100. So what we've done here is we've taken the mutant var, which is 50% allelic frequency, and then diluted that with the matched and the original, the wild type that was used to generate the mutation. And here we have BRAF V600K, and we've used the wild type to make a 10% dilution, 5%, 1%, 0.5%, 0.1%, and 0.05%. And what you're looking at on the left graph here is along the x-axis, this is the allelic frequency that's predicted or expected by our dilutions we've made, and then on the y-axis this is the V600K that the digital PCR called and read from the system. And what you see is this very, very tight linear regression, where the 50% is called at 50%, all the way down to 0.05% is called at 0.05%. Now, obviously, for reference standards in your hands, the 1% is probably the most is as far as you'd ever need to go right now, because the clinical relevance below 1%, is not known right now. However, for us, it's important, and we always see it as a critical part of our process that we're able to show that we're able to, the accuracy of our validation methods is down at this level. And likewise on the right-hand side, this is where we're taking KRAS G12D and done similar dilutions from 50%, and here we went down to 0.1%, and again you see the same linear regression. So it gives us great confidence that the genomic DNA standards, not only are we able to generate the standards of 50%, we're also able to generate precise standards with defined allelic frequencies. So moving on to FFB block production, we've developed a proprietary FFB technology, and this allows us to control very accurately the number of cells and cores that we include per block. It also allows us to generate a block with homogeneous cell distribution, so that every single section that we generate has the same number of cells within it. We can change and tweak the allelic frequency, and we can then control the section thickness, and of course the DNA content. So here, this is sort of a workflow of what we would do to generate a BRAF V600K 5% block and sections. We would grow up the cells, we'd mix the cells 1 in 10 dilution, would prepare the block and sections, would then cut the sections, and then evaluate the sections using digital PCR, and then would have our sections at 5%. What we then do, and what we've done to demonstrate and show the consistency of our blocks. So here is 8 independent blocks that we've made, and we've done H&E stains, and these are representative H&E stains from within the blocks. We've then taken so here we've taken 20 sections throughout the block and run DNA extraction on all of the sections independently using a Promega Maxwell platform. And we're very pleased with, and we think it's fantastic, is the level of consistency between the blocks, and also between the error bars is very tight. So within a block, each section contains almost identical amounts of cells and DNA content. We then took sections from throughout the block, and so here we have what we've done here is we've made 4 different blocks for BRAF V600D, where there's a 25% allelic frequency, 5%, 3.5%, and 1%. And what we did was we ran this to digital PCR, and what you're looking at here is the 25% block comes out dead on 25%. The 5% actually is slightly higher, and it sort of rounds up to 6%. The 3.5% is dead on 3.5%, and our 1% comes out at 1.4%. And what's really really encouraging for us and gives us great confidence is that once we've made a block, the allelic frequency throughout the block is consistent and is always maintained. And likewise with a BRAF V600K, we've made 3 blocks here. We've got 50%, 5%, 1%. 50% hits 50%, 5% is on the 5%, and the 1% comes out at 0.8%, which is for us it's close enough to 1%. And so we're really able to demonstrate and show that the FFP blocks we make contain the same number of cells and hence the same number of DNA that you can extract from them per section, and the allelic frequency that we define is consistent throughout the block. Here's another couple of examples just to show you for KRAS, where we have 50% KRAS block and 5% KRAS block, and the allelic frequencies come out dead on what they should do. So what we thought would be an interesting project to run was we developed a FFP block with a low cell number, and so each section had a total had sufficient cells to be able to extract around about 250, between 200 and 250 nanograms DNA. We always find that the theoretical DNA content is always going to be twice as much as what you can actually achieve due to the formalin fixation and the decrease in the efficiency of extracting DNA. And when we looked at different kits, we find that different kits will give you different total yields from the same sections, and also the DNA concentration can be impacted as well depending on the kit that you use. What I think is important and useful for molecular labs is always to understand what the kit, what the properties of the kits are, and what we find in our lab is that the column-based methods, like the QIAGEN column-based methods, are more variable between different people, but often they're very consistent within the same person running the same kit. Now, just going to move on to some external data that we have from our partners. So this is a diagnostic company that we're working with, and they took 3 of our BRAF V600E FFP blocks, and what they did was, so we generated this is using our BRAF 25% V600E block, the 3.5% and the 6%. They ran it on their 454 platform, and a completely independent, so this is an independent lab with an independent system, and they get dead on the 25%. When they run 25%, the 3.5% is 3.5%, and then the 6% is the 6%. And what's really nice is you can distinguish the 6% from the 3.5%. And so when this type of data really confirms that the degree of consistency and homogeneity that we have, and we claim using our validation methods, can also be validated using other people's methods. Similarly, here's another nice example where we're working with Molecular MD, and they took our EGFR T790M DNA starter pack, and this is where, so a starter pack is where we provide a mutant vial on its own and a wild type vial, and you can combine the mutant and the wild type to generate a dilution curve from, say, 50% all the way down to 1%. And this is exactly what Molecular MD did, and their curve was from 50% down to 1%, and they tested it using their digital PCR assay. And if you compare the predicted numbers of EGFR T790M copies per microliter to what they actually read in the system, it's incredibly similar all the way down. So it's nice. It's very nice to see that the stoichiometric dilutions that we achieve in-house can be achieved using an independent assay in a independent and external lab. This has been a nice study that Entrogen undertook, and this is using their kits. They have KRAS, BRAF, and AKT kits, AKT1 kits available. And what they did was they took our FFP samples and generated a dilution curve using our FFP samples, and were able to show that for all of their assays, they have a limit detection down to 0.5%. I should mention that all of our application data is available on our website in the download section. So if you want to read a little bit more about this study, you can go on there and download that. Now, I mentioned this at the start. I mean, we've worked very, very closely with the Proficiency Schemes to really understand where the gaps are in molecular assay testing, and we're officially working with UK NECAS and the European Molecular Quality Network, and are working unofficially with a major US proficiency scheme provider, as well as 3 others around the world. And I mean, we're always open to helping and providing samples for additional schemes, and so if any of you are a part of a scheme, then please feel free to contact me afterwards, and we can discuss whether or not we can generate custom blocks or custom sections or even new cell lines for you to help you with the proficiency schemes. So I'm just going to change gear a little bit for the last part of my talk, where I'm just going to introduce some of the newer standards that we have in our portfolio. The quantitative multiplex DNA reference standard is our latest product that we released at the end of last week, essentially end of February. What we have done is we've taken 8 of our genetically defined mutant cell lines and mixed them all together with precise DNA dilution to create, and what we did was we mixed them at the DNA level and to make a quantitative multiplex DNA reference standard, and then ran digital PCR analysis on this. And what we've done is we've covered 11 mutations originally, and this is through the genes of BRAF, KIT, EGFR, KRAS, and PI3 kinase. And the allelic burden ranges from 1% all the way up to 24.5%. And the idea of this is that you can now use this standard to start addressing what the variability and the reproducibility may be on multiplex assays such as NGS or Secronome. Our first beta testing labs took our standard and ran it on their ion torrent platforms. Here's the data that we obtained with our digital PCR platform on all of the mutations, and then here are our 3 partners using the ion torrent and the AmpliSeq panels. What's interesting, what's very gives us great confidence in this reference standard is that there is a lot of consistency between the panels for the majority of the mutations. There are some there's a few interesting results that were expected. So, for example, the deletion of EGFR is often not detected using some of the next generation sequencing platforms, and at the 2% level, it wasn't detected using the hotspot panel V2, where it was detected on the cancer panel. Similarly, the T790M at this low 1% level, it couldn't be detected on these panels. There's a few other. So there's this lab here was unable to detect the G12D, whereas the other 2, not really sure why. And then there's a kind of another mutation that gives a slightly unusual result, is the PI3 kinase E545K down here, and this is likely to be attributed to strand bias. And the feedback we've got and what we are looking to do next is really, now that we have this standard, we can really start to understand where there is variability in these assays, and use an external system, using a digital system, to really show where tweaks or improvements may need to be made to the particular panels to be able to detect these mutations. And what we're now doing as well is, so this initial offering has we've validated 11 mutations, and what we're going to be doing next is we know there's a number of SNPs and indels across these genes within our cell lines, and so what we'll do is now validate the indels and the SNPs across these genes, and then once we've done that, we will then be able to add those additional features to these 11 already, so we will then be building up a multiplex standard that will contain somewhere in the region of 30 features, and then after that there's another 20 that we'll be focused on. So the aim would be to have a multiplex standard that would have up to 50 features. So just as I bring the talk to a show and to an end, I will just introduce very briefly the kind of the more other reference standards that we're looking to develop, and always open to new collaborations or ideas for reference standards and reference materials. So we've got in situ hybridization reference slides coming in March 2013, and what we've focused on initially are translocations for liquid tumors, where we developed interface spreads onto glass slides. And then we also have used our FFB technology to generate FFB sections mounted on slides for our P10 deletion, PAX3FOXO1 translocation, and the EML4 ALK translocation. Here's some preliminary data just to give you a flavour of. We're able to use our standards to really test how well different probes work on the different translocations, and the feedback we've had from our beta site is that these slides will be an ideal development training tools for any new ISH probes that are being developed, as well as being able to being able to use as a batch-to-batch testing to ensure that the probe and the user has run the assay correctly on that particular day. And of course, because we've generated the EML4 ALK FFP slides, it's then pushed us towards looking at how we can apply that into immunohistochemistry. And so what we are now doing is developing a method for being able to provide reproducible and finely tuned range of staining intensities for this area and a number of other targets. So just to summarise, this is a list of almost 40 mutations that we now have available for as quantitative molecular reference standards, and we can precisely manufacture allele frequencies from 1% all the way up to 50%. This is a slide that we often include at the start of our talks, but I thought it was more appropriate to put it towards the end. I mean, in building the quantitative molecular reference standards that we have, it does allow kit developers and kit manufacturers to optimize new kits for the targets that we've spoken about, but it also gives labs the molecular pathology and molecular diagnostic labs to really be able to test the limit detection of their assays in validation studies, as well as testing on a batch-to-batch run to ensure that molecular assays are working on the day that they're being run. And as a team at Horizon Discovery, we do believe and we strongly feel that if we can eliminate molecular assay variability, then this will ultimately lead to better patient outcomes. And so I'd like to thank you very much for your time. Please feel free to email me any questions after this talk, and I'll be very happy to answer them.